Hi, folks. I'm Misha St. Amand. I'm the head of marketing here at JuliaHub. And today I'm happy to welcome you to our latest webinar, Genomic Data Analytics with JuliaHub, SingleCellProjections.jl, and GLMakie. I'm here with Phil Burns, who is a sales engineer with our team, and I'm going to let him jump off and share this presentation with you. Thanks for joining us. Phil?
Thanks, Misha. Welcome to the seminar, and my name is Phil Burns, sales engineer here at JuliaHub. And if you notice, that name is Rasmus Henningsen, who is not me. He is the developer of this package, and these slides are from the JuliaCon conference in July. I highly encourage those of you who did not attend to attend. It is truly an excellent use of your time, and you will profit from it, I assure you. In any case, the objective here is twofold. On the one hand, I want to introduce you to this package. It's a wonderful package. It's freely available. It's open source. On the other hand, I also want to illustrate some best practices when programming in Julia. I won't go too much into detail, but one of the reasons why this package works so well is that it was built with the canonical design patterns of Julia, namely generic functions and multiple dispatch. So the developer of this package did an excellent job. And so it's not only for solving this specific application of sequencing and representing sequenced mRNA single cell samples, but also for writing good solid Julia code. So what's going to happen in the seminar is we're going to actually go through 3 different tabs. And the one I'm going to have the slides from the JuliaCon presentation, my live demo of the code, which is here in the Pluto notebook hosted in JuliaHub. And then at some points, we'll actually look at the GitHub repo very briefly, just superficially, so I can illustrate for the viewers here why it's well done, why there are particularly very strong patterns in this code. So this is the source code. This is from the GitHub repo of SingleCellProjections. If you were to go here, that's where I am right now. It's under BioJulia and SingleCellProjections.jl. So if you go to it's sitting in the BioJulia home. Anyway, let's get started with the basics. So you're all familiar, presumably, with the single cell procedure and sequencing. I'm going to skip through this. This is actually what's very important. Oh, by the way, if there are any questions, feel free to interrupt. That's actually really important because things here build consecutively. So if, for example, you don't understand this slide and this data structure, nothing else in this presentation will count. So Misha, if you see any questions, let me know. I'll wait a minute or 20 seconds.
Absolutely. Folks, feel free to type in that Q&A. We'll be using the type in features for questions. So don't hesitate to do that.
Yeah, specifically for this slide. I'll talk about the slide, and if it's not clear, I want to elaborate further. I'll illustrate it in the live demo, but I want to make sure that the big picture makes sense to people. So this is the main data structure that we're going to encapsulate in SingleCellProjections.jl. This is our data matrix. We call it a data matrix. Now, the developer of this package created a custom type. This is a thing that's very important in Julia, is to use the right type. So created a new abstract data type called a data matrix. This abstract data type is a high-dimensional data type, meaning it's not just rows and columns. These are your genes, right? These are your columns, and these are your rows, your cells. But each of these genes themselves, each one of these, has information for each single one, and that's also stored in the data matrix. What that means is that I can actually pull from just this gene alone, right, information about that gene sample, about that gene in this sample. Right, so we can almost think of it as like metadata or nesting of data. All these numbers here only represent counts. So if you look at it, you say I have a gene and I have a cell. This is a sample, right? This whole thing's a sample, and there might be multiple samples in my data sets. In which case, you have a sample ID like P1, P2, etc. In this case, let's just not worry about that. Just say it's from the same sample. So I have my cells in the sample. I have my genes in the sample. And I have my counts. Now, what's really important is that most of these are empty. They're not zero, they're null. That's it. So we're going to have to use, and so this will also impact the design of the program, because considering that most of the of this matrix, and this is a matrix of counts, we can call it a matrix of counts, is empty, we obviously need to use a sparse matrix data type, which is what we end up doing. So when we will evaluate the value of the data portion of this matrix, a count data portion, you're going to see that it's actually a sparse matrix, which means that it's going to be represented and computed very differently than if we would usually see as a dense matrix. And part of the advantage here is by computing things differently, whether it's a dense matrix or a product of matrices, by discriminating on the type of matrix or matrix expression or sub-expression that we're looking at, we will do different things. This is where the multiple dispatch and high performance comes from Julia. So again, concretely, in the data structure that you're going to see with respect to the count data for a given sample, you're going to have each column is a cell, each row is a gene, and you have the counts here. You will then furthermore have a separate table for each cell and for each gene, also in that data matrix object.
Bill,
So,
just with a quick question, what do the counts represent?
They count. They literally count the frequency counts. So there's like the number of times it's been counted. So this is the number. It actually says number of detected molecules for gene I and cell J. So correct. So specifically here we have RPLP1 in cell 1 has a value of 60, right? And MALAT1 in cell 1 has 376. This is literally just count data. So it's just counting. So you could infer that these null ones are zeros in that sense, but we don't do that obviously because it would break our sparse matrix structure. Are there any other questions, Misha, before I can go on?
That's the only one so far.
Okay, good. So let's go on. I'm sure some of you are familiar with this, but what I want to do actually is quickly show you a little bit of code, and then I'm going to jump to some actual code. This is going to be loading a count.
I spoke too soon, Phil, before you move on.
Go ahead.
Which is data filtered in this?
Oh, yes, absolutely. So this is great. Okay, this is obviously someone who's done this before. Yes, we absolutely have the canonical data flow. We absolutely do have the canonical data flow. So I will get to the canonical mRNA sequencing data flow once I demonstrate just the high dimensional structure of the data matrix. So to get this count data into a data matrix object that this
Sorry, one more.
Go ahead.
Is the count expression level? So I'm sorry, excuse me. Let me rephrase that. So the count is expression level. Is the question.
I don't know what that means. I don't know.
Okay.
I don't know what that means. Anyway, so we can follow up, async after on that one.
Yeah, guys, folks, if there is a question we can't answer or we can't do, we will have somebody follow up with you afterwards. So don't get frustrated if we can't do it right now.
Surely be me, but we'll see. Anyway, how do we get this counts? Like, let's start talking code with this package. Well, obviously we have to load the package, and now I'm going to jump. So I don't want anyone to get too confused. I'm going to jump to a Pluto notebook, and to actually load the package, and I'm going to skip this verbiage because you don't need it. I'm here to talk to you. I first add my SingleCellProjections package. I add the data frame, CSV, all the things I'm going to need to work within a Pluto notebook instance in JuliaHub. And specifically, I'm going to be pulling from 3 data sets. Now, the data sets I'm going to be using are actually the same data sets that you can get from the tutorial. Yes, there is a tutorial if you go to the GitHub. So like I said, you need 3 tabs open to do this, but because you can stop me and ask questions, it'll be manageable. You can go, if you go to the GitHub, you'll find a link to the tutorial. I am using the data sets from this tutorial. I just use the cell annotations, right? So those are the annotations that I spoke about here. This cell annotations file, what does this mean? That means stuff about the cells and the stuff about the genes, okay? It does not include the counts, all right? The counts are in a separate file. Those are the sample counts, right? These are the P1, P2. These Ps, each one of these Ps represents a sample. So those counts are in these samples, okay? And the annotations with respect to the cells and the genes are in this file, all right? Just to be very explicit. Misha, any questions right now about that? Nothing? I'll keep
Nope.
going. All right, you're good, I'm good. So I'm gonna start working with, I wanna show you actually in real life how this load counts works. Yeah, I have I have to provide a path and a file name. Now, in the case of here it is right here. I have my load counts function, and I define my sample path with by defining. I just define an absolute path explicitly with the file name. So it's essentially a list of paths, and then I also define the sample names P1, P2. I'm only loading 2 samples. And if you see what it returns a data matrix, okay? And it has 33,766 different genes, variables, and the observations will be my cells. So I got 35,340 cells. It also shows something very interesting too, is that I see this sparse matrix CSC. That's the type of the counts, okay? So if I were to look at the counts type, if I were to do, let's see, counts. Yeah, if I were to look at the type of counts, right, it's going to tell me that I have a sparse matrix for 64-bit, 32-bit integer, data frame. So that means these two, the annotations are actually data frames. So if I were to do something like counts dot var, that should give me a data frame. Yeah, voila, gives me a data frame. You
So
see?
a question that just came in a second ago.
Yep.
Can I load 10x genomics cell ranger output?
It's a good question. Yes, you can. It's just been added. So that's been added to the package. You have to go to the GitHub repo. Was that an issue recently? We can follow up after that. I think that was just added recently. I think he added as a feature recently, but I'm not 100% on that. So, but check the repo. If it's not clear, contact me or the developer. He's actually a very accessible guy. So this is a concrete example of the annotation, right? So we have 2 data frames in this data matrix. Again, we have this variables data frame and the observations data frame, and these are both data frames corresponding or that were built from the cell annotations, whereas the actual count data sits here. Just to reiterate. All right. So, and I can also do something like this, like counts.obs, right? And this is should give me my observational data. Now you see it here, right? You have your gene, you have sample name. You scroll over here. Now, for those talking about filtering and things like that, this is definitely like where this will happen, right? Looking at the mitochondrial contamination. All right. Let's see. I want to go back. Oh, and lastly, I can show you the data itself. I talked so much about the sparse matrix. This is the counts. Okay. So like if you were to try to go in and say, no, I want to look at the counts data, not quite, right? We're looking at, this is a column major sparse matrix. You know, it's encoded differently. It's really not designed to just be queried directly, queried in the same way that you would say query a matrix or some sort of like nested array or something. It has a completely different intention. And that's consequential when it comes to the computation side. And I'll show you some benchmarks from the JuliaCon slides that illustrate like just a little bit of like forethought on the developer side and working with by using generic functions and multiple dispatch, he was able to make this thing fly, especially compared to Seurat specifically. We're really comparing to Seurat, and it's really a lot faster. Okay, I think I've also illustrated, and this is just handy, just like a bookkeeping thing. Like when you have a data matrix of any kind, you can actually do a trick. Like if you're in Julia in general and you see some custom object, like here it says, oh, it's a data matrix type, you may not know what the fields are. Like, how did I know to do matrix or something? There's a command called field names. This is for the people who are not super Julia familiar. Do field names, and these are what called symbols, but these are actually the fields in your in that struct. This is actually a struct, And these are the fields in there. So you can query any of these. And again, it would just be something like, that's actually a type. So I need an instance. I could do counts dot, models, for example. And I'll get that, which right now it's empty because I haven't done anything. And what you'll see later on is one of the reasons why this is called single cell projections is that we accumulate transformations in this models field. So why does this even exist? I told you why the matrix exists. This exists to keep track of the counts. These two are observational data. These are ID columns for processing. That makes sense. What's this models thing? The whole point of this models thing is that as we go down the mRNA sequencing process, as the users here are probably familiar, there are things you do. There are transformations, normalizations. Well, actually, let's do it in order, right? Filtering, normalizations, transformations, or maybe it's backwards. But anyway, you do things. You change, you mutate your data, and you want to make sure that you're capturing that. Like, you don't want to be losing that state of how did I make this? One of the advantages of that is when you're trying to say, okay, I don't need to store it in a new variable. I don't necessarily need to rerun the process. I don't need to have multiple objects in memory. One of the things, and part of the things that motivated this models field, was the concern that you would have to have, say, progressive development of your, as you're working through the sequencing process, you're going to have several, these hierarchical nested data frame type things sitting in memory, taking up a lot of space, and that's not good. So you're able to basically make it easy to reproduce and track the transformations and changes you made and your progress using this, what they call the projection model, where it is derived from the concept of a projection in vector spaces, but that's not the primary concern. So I think now I've completely, aside from showing you the source code, I think I've very fully illustrated the object that's created when you execute this load counts function. But it's very important because once you understand this data matrix object, you can work with this on your own. You can make your own changes, etc. So let me see. I'm going to jump back into the slides, and Misha just flag me if there's a difference. So we've done this, and then we're going to move on and start doing some transformations. Is there any questions right now or not yet? No? Okay, I'll keep going. So now we're going to start doing the workflow, right? And in this case, what I have is, this is just me showing what's going on, is actually, I actually do it a little differently than he did in the JuliaCon presentation. Is first thing I do is I actually join the two tables. So I actually end up joining the counts data with some of the annotation data, for the simple reason that I want to be able to filter on the mitochondrial contamination and some of these other features, right? So there are people who asked about filtering. Well, here we are. We're going to start filtering. But in order for me to filter, I have to get these annotations into my count structure somehow, right? So he actually, one of the functions he has is precisely that, where it says var counts underscore fraction, and literally and this is only for Pluto. We have a counts new in here. If you do this in your VS Code instance or something, you would not have this. This would just be this counts new would not exist. This is just for Pluto because Pluto's reactive. If you have questions, we can discuss later. So what this does, it's going to create a new data matrix object. It's going to be a copy, basically, of what this thing is. And what this thing is doing is it's mutating the counts data matrix by adding things from the annotation, right? I want stuff from the annotation. I want this guy, right? So you can see here, there's actually a regex, and I want specifically, gene expression, and I also want to get my fraction MT, and this is very important. And it tells you also here from where to get your annotations, right? It says, all right, I want to get it from this specific file, which is the one I mentioned before, and then they're joined. So when you end up doing that, if I look at the new file, so if here's a new file where I have the it's basically a massive new object. I can now start filtering on specifically the mitochondrial contamination and the NCOUT for the ADT and an RNA. To do this in Julia, I'm going to use not the usual plots.jl library though. I'm going to use something called statsplots, and statsplots.jl gives me this method. It gives me this thing it's called a macro, this little %df. What that literally means is that I can actually directly reference the field name in this expression without having to put the data frame itself in there, right? So I can put the field name directly in here, and instead of having to struggle with, well, I have to specify this whole counts new.obs and put it in here. No, I can just put the data frame itself, which is this observational data frame goes out in front, and then I have the function here, and I just specify this. This is just syntax, but I want to be very clear about the syntax. Is that the data frame that I'm using in this macro, which is called a macro, which is metaprogramming in Julia. This is the observational data frame for counts new, and I use it several times because I'm going to define three violin plots, because I want to show obviously the number of genes in the cell, the molecules, and the contamination, right? To get an idea of of what I should be looking at and what I'm filtering. Does this make sense? This little
I have a couple questions, not quite directly related to that, but the first is the matrix is sparse with several empty spaces. Does that mean the counts are 0? If not, how can we use the data for modeling? What does it tell us about the relation between the gene and the cell?
We'll get there. We're going to get there. This is all built into the package. Go ahead.
And after that, there's one more. So.
Okay.
There was a request to elaborate on the modeling. How is it done? Is it PCA-based embedding or epoch or something else? How do you essentially take care of the batch distinguishing?
Oh, that is a detail I don't know. I know how the I know how the expression is evaluated in the matrix. I would have to go through the actual detail of like the execution, but that I don't know. So that's a good question. That's very technical, unfortunately. So I don't know that. Let me jump back into, okay, I'm going to finish these plots. I'll show how the plots are developed, and then we're going to move on to, and another thing too, which I'm very aware is I know people do this differently. Like there are people who have different workflows or like, oh, why, I get it. This is just to illustrate one simple approach. I know there are various opinions on how to do this. So again, we're using the statsplots.jl library here to generate these violin charts. The next thing I show are also scatter plots, and I'm not going to belabor this. I think the audience knows why I'm doing this, but basically I want to show, okay, like where should I set a cutoff? I mean, basically, if I want to start thresholding values here and get more, try to count more meaningful data, where does it make sense to threshold? One way I do that is I define a filter function, which is a little different than the way you would do it, I don't know, like in R or something with slicing. Here, or Python, like you don't really slice here. What you do is you define this like filter function, and I just define these criteria of, say, in this case, I wanted 5,000 or more unique feature counts, which was inspired by this. Just I was just looking at like linearity or something. I kind of made it up. Fewer than 1,000 unique feature counts. Okay, so anything below here would be truncated, and under 10% contamination. Okay, so everything above this 10% would be excluded. That was the filter logic, right? And then what I do is I return the Boolean the AND combination of all these, right? So it's this expression and this expression and this expression all evaluated as one consecutive expression. Fine. Well, how do I use this? So again, he part of the package is he built a filtering helper function called filter_obs, which is meant to filter with respect to observational data, right? Like we're working with observational data, so he built that. And what you do is you put the fields in which you are addressing, and it uses kind of strange syntax, right? Like it has like this like list of symbols, and then it has this arrow with the filter logic, and then it has a data frame name, right? That we're filtering, and that is the syntax for the filtering here. So you don't need to, when you're doing filtering, whenever you define your filtering, you don't need to write your own custom filtering function per se. All you need to do is define the filter logic. So you would take the filter logic and then inject it in this as an argument here, rather than like writing a filter function directly. And there's actually a really important reason why you need to do that, because as we remember, this is high dimensional data, right? Like it's not simple. It's not just a 2D matrix or data frame where you can just filter things. Like there's observational data associated, and there's annotated data associated with every entry in that count matrix. So if I'm giving something up, I'm actually changing the associated annotated data, right? Which actually matters a lot, because if I'm looking at, say, contamination or other properties of cells, for example, if I'm looking at cell properties, and it just so happened that I filtered that whole cell out, I might be, and I don't account for those changes in my filtering, right? I think people understand what I'm talking about. I gotta keep my my debits and credits, so to speak, consistent. And this just shows a plot of the filter versions, right? And I suppressed the code, but it's right here. And as you see, again, we use these @df. This is the macro from statsplots library, okay? This is, again, the data frame name. Now, this is the filtered data frame, right? Filtered, and this is the observational data from the filtered data matrix. So I have the filtered data matrix from as the return value from this filter observation function, okay? And it's exactly the same data type. It's still a data matrix. It's no different than counts, right? Counts was a data matrix. It's the same field names. Likewise is filtered. And that's also important too, because when you're doing the, I don't know if we have time, but when you do the, when you run the code, you will have multiple dispatch on type, and he's able to use, because he did this very well, we are able to use the same generic code throughout across the entire process. So while we're filtering, while we're normalizing, while we're transforming, we're able to still use the same exact API for the data matrix object. So it's very I think logical abstraction. Okay, so here we are with the plotting of the scatter plots, right? So we have the stats plots and scatter function. I don't know if anyone has questions on this syntax. This is really a thing from statsplots library. It shouldn't be super complicated. It's just the only thing that's weird is you have a macro here, but again, you just put the data frame here first, and then you put the statsplots.scatter, yada yada yada, and you'll have the symbols of the things that you're plotting your x and y axis corresponding. And you can customize the labels and title. I think it's pretty straightforward. And you can see the effects of the filtering, right? Like this is before the filtering, and this is after the filtering. So I'm truncated now. So now there's nothing above 5,000. There's nothing below 1,000. And likewise, I truncate here too. I threshold at 10%, so that drops down there. Okay, so that's all this is showing, is that the filtering worked. Okay. It's also interesting too, because as I mentioned, like when you filter, you're going to have different, like you're gonna like remove stuff possibly, and that actually happens. So like in this case, what we're specifically changing, we don't change the number of genes, but yes, guess what? We do lose cells, right? So because we lose cells, again, if we didn't keep, one of the main tasks of this package is to keep correct count of annotated data, so that we don't double count or miscount somehow the annotations as we transform or remove count data. So yeah, so you can see it here very explicitly. It's like the gene count is consistent, but the cell count has changed. Okay, it's obviously gone down since we filtered. And lastly, well, not lastly, but from the filtering perspective, one thing I do want to show you is this. I talked about the models for the data matrix object. Like, why does this even exist? Well, here you can see it right now, right? You can see exactly what we did. When we started out, this was our initial construction of this object, right? Here's how it was constructed. Here's a sign. Here's a column. Okay, we have this the control column. Fine. So what? However, now we applied a filter to it, and we keep track of it. So now we see step 2. There was a filter model, and specifically, we were looking at these 2 fields, and the filter itself that was applied was the filter logic function that we defined, up here. So this is where the projection part comes in. Part of it is the ability to keep track of every single one of these incremental transformations, and that's really important in this work. And if we contrast that like with this one, right? So this is the count.models, right? You don't see that filter model step that we just did, the filtering step. It's not there because we didn't do it. Okay, is everyone good with that so far?
I'm not seeing any questions.
All right, cool. Let me see if I can get away with jumping here a little bit. So you've seen some of the work, right? His workflow, I do filtering before he does transformations and normalization. We'll do that too. One of the things I want to show you is that, I'm jumping a little bit ahead, is that you can also do t-tests here. There are a bunch of statistical tests that you can apply to count data across samples. So, for example, as you've noticed, like in our example, we have P1 and P2 samples in the same data matrix. You have the option of obviously testing across that. And these tools are nice. They're handy because, one, they work, but two, they count for having multiple samples in the same data structure. So you don't have to worry about like breaking it out into samples, right? It will automatically look for the quote unquote sample name. It's going to look for P1, P2. It's going to look for it's going to do the t-test correctly. So, obviously, for a t-test, you'd want to have just any of these hypothesis-related questions. You'd obviously want to have, unless you're doing an f-test, but one versus all. But if you do like a t-test, you obviously want to just have 2 samples in your data matrix. So just keeping that in mind, that you do have, in addition to the filtering and the normal procedure, you do have statistical tools available to you for your analysis, and they're done within the same API. So you don't have to import any packages. You don't have to like change anything or worry about bookkeeping again for across samples. That's taken care of. Let's go back to the demo. All right, cool. Let's see here. I did some analytics here, but I'm going to kind of sink of time, skip over because I was just basically demonstrating, how things have changed across the filtered and original data matrices. So the next step is the transformation, and the transformation that we use is actually technically an approximation to the negative binomial, for the simple reason it's a lot faster, as you can imagine. So we end up estimating a Poisson distribution, or fitting a Poisson distribution, estimating a Poisson parameter, because we're going to fit a Poisson distribution, and then we get, and then doing that, we can compute the dispersion. And so what ends up happening is that when we fit this transformation, you'll see something. You're going to see a theta parameter. You're going to see a theta, beta naught, and a beta 1. And I assume the audience is fairly familiar with this. If we scroll down to here where the transformation is complete, and the function that's used is called SC transformation. This name is actually derived from the famous R package. Okay, it's basically the same, right? It's the same. And where is it? Right here. Here you go. So here are your fit. Here are your estimates. Okay, and so for these genes, we have these estimates. So that's your transformation. Now we have this wonderful transformation, but the question is also, how does it look? Before I look at the charts here of what the transformation does to the count data for the genes, I also want to look at the actual expression itself, because this is really important. We created this transformed data, again, another data matrix using the SC function. But it's interesting, you see this like A plus B1, B2, B3, right? Now, why is he doing that? What's the point of this? And the point is that this is to communicate to the user that this is actually not evaluated. So this is going to be using lazy evaluation. This package does not go ahead and just necessarily compute this expression for the simple reason it's slow. So the package waits until you absolutely need to do it, and then at the moment where you have to make a decision about how to compute this, it will actually implement a dynamic program. And the dynamic program will look at the actual matrices involved and say, okay, this one is upper triangular and this one is symmetric, so this has a cost function of that compared to this approach for evaluating the same expression, right? It's going to look at different ways to evaluate this expression and choose the one that has the lowest cost. And it does that using a dynamic program. And then once that lowest cost is found, that's the method for evaluating it. So it's this dynamic program that will happen downstream, right, later on, is why we have this like suspended lazy evaluation thing. Like, why is this even here, right? Like, if you look at the original, before we did this transformation, there was no thing. We just had a sparse matrix, right? That's it. Now we have this like suspense, right? We have an equation. We have AX plus B1, B2. Okay, so I have a product of matrices on one side, and I'm adding this A in the other. Okay, right? Well, it hasn't been evaluated yet. And that's, again, a very, very key feature for performance advantages. But I am jumping ahead a little bit here, is that when this gets evaluated, there's a dynamic program invoked that looks at the various options when it comes to branches across all the computation options or evaluation options, and depending on the structure of the matrix and what it sees. And you can look in the code, actually. Now I'll jump. If you actually want to see the logic under the matrix expressions, it's under the joint sparse chain. This is where the dynamic program is executed or developed, right? So like this is a key piece of code. It's a joint sparse chain.jl. So when you're looking at how this works under the hood, it's here. It's going to be this one. This is where you really build out the dynamic program to decide what to do. Okay. So this is going to be a funny thing, right? Because again, we went from a thing that's like tangible to this expression thing. And actually, if you want to look at the typing itself, you could go here, and you can actually even see that we have two structs here. We have a matrix product, right, and a matrix sum. So what I'm actually going to do is you're going to see that the matrix product corresponds to this, which makes sense, and the matrix sum is all of this, right? And this is just a matrix. So there are types for this expression precisely so we can evaluate it as parsimoniously as possible. Anyway, we'll keep going. So if I want to actually look at the type of this A, I just did a type of command. It's a matrix sum for the whole thing, as I said before. The terms of the matrix sum has two, so it breaks it out into one matrix and then this product of three other matrices. And if I keep drilling down, I start getting the specific typing. Right now it says, okay, this one is a sparse matrix. Yes, finally you teased it out of me. And this one is just product of matrix things. Okay, there's not really that much more I want to do with that, right? But this actually is kind of important if you actually want to go into, like, if you want to stop and like I kind of do that here. If you want to stop and like evaluate something or examine it, you have to know that you have to break this down, right? You're not really meant to do that though, but you can. But if you're going to, just know that the typing is a little novel. Misha, any questions or not yet? I'm just gonna keep going. With respect to visualizing the transformation of the counts after doing this, the negative binomial approximation with a Poisson, I just do that right here. It's very straightforward. But what's interesting though is for me to actually count it, right? Look what I did. I actually did, to make this visualization, I actually kind of hacked the API a bit to pull down these values so I can actually make a sum, because I wanted to be able to take the sum and actually sum across all the cells, and which is what this does, is sum across all the cells to see, okay, what happened to the distribution of the count data? Like I want to see, did the transformation that this something, did it make it more tractable, right? Does it actually look better? And I guess you could say, yeah, sure. I mean, this definitely looks better than this. So I mean, this is like, I don't know, crazy fat tail or something. This definitely looks more tractable than that one. So that was the whole point of this little bit of code here, and then plotting it. And the idea is that I was able to get into this object by actually using this compute method as well. And I was able to use this compute function for the single cell projections package. This is again multiple dispatch, but on A turns 2 is this matrix product thing, right? So that means if I'm going to go just click the source code, so I'm looking at the matrix product. This is this guy. So I want to look for a dispatch on this, which would be here, and boom. So like this is the function I'm looking at, right? So if I'm going to be dispatching on A terms 2, which is this matrix product thing, that means I'm calling this guy, right? This is the matrix product. And then you have one here with the matrix sum, which is for the whole expression. Anyway, I'm getting extremely technical here, but for those of you who want to be able to recreate what I did or just want to get really deep into the code, it's pretty accessible. But I hope you have enough orientation now to figure it out. This is not terribly enlightening. The last thing is the normalization step, and then we're going to do some PCA and SVD. But the normalization here is we're going to just do centering and a regression, right? And we regress against we regress out, I should say, the mitochondrial contamination. And then finally, we are going to filter by the cell type again. So we do another filtering. And if anything that's an other is removed. Now, here's what's really cool is we've done a lot of stuff to this data set, right? So we have filter2.models. Let's see how that works. And click here. Look at all the stuff that's been done, right? It keeps track for you. And if I look above here, normalized models, you can just do the comparison. It also includes the rank of the transformation, which is actually really cool, the linear transformation. So, and then likewise, when I do the PCA, if I can do the same thing as well and do reduce.models, and that's going to give me the final stage right before the plotting. So 20 degrees of freedom. So what's actually really neat is I don't need to necessarily just like walk you through this, like because this is so neatly encapsulated, or there's such a nice API. It's nice that I can just summarize it, so I don't have to go back through my code and try to recreate it or remember what I did. I can just query the models field for the data matrix. So it's I personally think that's really nice. And lastly, we do a plot. We use GLMakie as the library. The reason I use GLMakie is for the simple reason that it's interactive. Okay, so hopefully this works reasonably well. I have to give it a second. Yeah, GLMakie is great. It lets you drag. It's three-dimensional and it's interactive. And this is going to make a very cool plot, a three-dimensional plot that we can scroll around. And yeah, here we go. And this shows some pretty nice clustering. So that's, I would characterize that as quite nice. Now, there are other options too. I'm just going to suppress that for my browser's sake. You can also use, I could use this UMAP representation, and I could for reduction rather than SVD, and I could plot that like clustering. That's another option. You can use TSNE clustering. There are a lot of options for that, and I could use all of these for plotting. So you have a variety of options with respect to clustering methods for the plotting. Let's see here. Now, if we go into the performance, quickly want to cover the benchmarks. So this is why people should be at least trying out this package. Is when it comes to the performance, this is for normalization and a 50-dimensional SVD. And here are the specs: 2,000 genes, 2 samples, right? 35,000 cells, 2,000 genes. So do the math in this product, and you have 2 samples. It's clearly a lot faster than Seurat, pretty much all the time. And the reason why is, and here's another one, similar, more genes this time, so more rows. And this time again, this is the light blue, or I guess purple. That's the green. It's the orange. It's quite a bit faster, right? It's substantially faster, time in seconds. This is actually kind of funny when we when we go to 20,000 genes. So again, we're multiplying by 10, right? Seurat just doesn't work. Actually, ScanPy does a decent job. That's because of the int32 encoding. We're using int64, or excuse me, float64. He's going to change, I think, to float32 at some point, and this will be much better. This will improve and will be faster than ScanPy. Oh yeah, this one. Yeah, ScanPy was a little bit faster. So but Seurat doesn't even work, right? So this is really great, and one of the reasons why, if we look at the memory usages in general, yeah, if we run this by default, this runs float64. If we run it float32 and we compare it to ScanPy, it's basically at the memory level, it basically tracks it. Seurat just goes up pretty exponentially. So you really have a scalable process here with single cell projections, which is again why I'm an advocate of this package. And he goes into some detail here about why it works. We can share this with you or post it so you can go into more detail. But remember, it comes down to matrix expressions, right? So the key thing I talked a lot about the lazy evaluation. Well, this is why, because we don't store the large dense matrices. And more importantly, we only need to compute the matrix product and the diagonal of the gram matrix. And it was like, why do we care about the inner product of columns, right? Well, because in this case, our inner product is the covariance, right? So we just care about the variance. So we filter that out and we ignore everything off the diagonal in the covariance matrix and consequently, like, we save a ton of computes, and we're able to very efficiently evaluate the matrix expression object that he created. And he goes into more technical detail here. It's really worthwhile, I think, if you want to go really into how he's justified his design. It's quite a long presentation. But the summary is that it's fast, it has the tests you need, and most importantly is that the package is developed. It's on BioJulia. It's part of the BioJulia package, and also most importantly is the developer is open to feedback and very keen to improve and expand the use of this package, which to me is actually one of the most important things. So I guess with that, I'm basically done. If there are any other questions on the floor,
I have a couple before we wrap. Where does this plug into the single cell analysis workflow, post alignment / feature assignment? Is this designed to be downstream of the cell ranger exclusively? For Python / R users, would you say just diving in with this package would be an okay place to learn using Julia?
That's a good question. I think I would love to say yeah. I would say just do a little bit of Julia stuff, because I'll give a concrete example of why I would say, there are many resources on just the basics of Julia, like this typing, for example. It's important to understand that why there's a compute A with this function, this multiple dispatch, right? You have 2 functions compute here, one for a matrix product and one for a matrix sum, right? And then one here for this diagram type that he invented. This should not throw you off too much. If this really throws you off, I would say we have a really good there's an MIT course on Julia by Christopher Caucus. That's really, really, really good. I frankly use it. It's on SciML, but the first 2 lessons are on Julia itself. They're excellent. I would just say, yes, you are free to jump into this, but I qualify that by saying, make sure you understand 2 concepts: generic functions and multiple dispatch. If you qualify in that, then you're safe. Otherwise, yes, you can use it, just kind of type along. But things like, there will be times though, like you want to do stuff like I did, for example, like when I decided to go into the data structure, into the data matrix itself, when it was no longer just a sparse matrix, it was now this custom type, right? This abstract type he made. Like, how would you wouldn't be able to do this, right? Like, you would not be able to like figure out how to like get in there and mess around, because sometimes you want to evaluate midstream what you're doing to check things. That's why I did it. Like, I wanted to see, yes, he did a transformation, but I don't believe you, right? show me. And that's how you do it. So I think it's spend the 1 or 2 hours on very basic Julia, just to learn multiple dispatch and generic functions. Just know that basic thing and some basic stuff about types, and that's all you need. Because then you can go into the you're not intimidated by this, right? Like when you see this as your data matrix, because remember, this is a data matrix for the transformed data matrix. Like if I look at like counts.matrix, right? I just get a sparse matrix. Like everyone, you're an R user or Python user, like, okay, MATLAB, whatever, like, whatever, that's easy. I'm cool with that. But then I hand you this thing, right? You're like, what do I do? And you need to be familiar with typing and stuff like that. And it's also important to like just know like when this function is firing versus say when this function is firing, because they're the same function name, but they're, again, there's multiple dispatch. Okay, so I hope that answers the question. If not, please follow up.
Okay, I think those are all the questions that I've seen. So I want to thank you, Phil, very much for this informative webinar. Really enjoyed it. I want to thank all our attendees for joining us. There were a lot of really great questions and a lot of great stuff here. So I did receive a couple questions that I answered. We will be sending out the recording in a day or two of this webinar. And in that email with the recording link, we'll include some of the resources that Phil's brought up today, as well as any appropriate links to things he's referenced. And for those of you who may not have had your questions answered, we will be following up with folks. So thank you very much for joining us. And keep an eye out on that email for future webinars that we'll be hosting as well. Thanks, everybody, for joining us. Thank you, Phil.
Bye-bye.